Hello everyone, I'm Laura Dormer and I'm the editor of The Evidence Base. I'm pleased to be joined today by Kelly Magee from Flatiron Health. Welcome, Kelly. To kick things off, please could you introduce yourself and tell us a little bit about your background?
Sure. Thanks, Laura. I'm really excited to be here today. My name is Kelly Magee. I am a nurse practitioner. I worked in the hospital setting for 15 years, taking care of people with cancer before coming to Flatiron a little over 6 years ago. At Flatiron, I have the privilege of working with a lot of very smart people on cross-functional teams of engineers, data scientists. We work together to develop meaningful evidence from electronic health record data. My role on the team here is really to be that clinical voice. So I provide the clinical context, ensure that we're developing data sets and research studies that are meaningful for patients and clinicians. I came to Flatiron because I was really inspired by the mission here. So Flatiron's focus is on improving cancer care and the outcomes of people with cancer by learning from the experience of every person with cancer. So really felt like an opportunity to have a much bigger and different type of impact than I was having in day-to-day clinical care.
You're speaking to us today about research being presented at the ISPOR 2025 conference on evaluating the reliability and validity of machine learning extracted real-world response data in solid tumors. So could you start us off by describing what we mean by real-world response data and why it's valuable to study in oncology research?
Yeah, I'd be happy to. And this is something as a clinician that's really near and dear to my heart. So response is really critical for patient care. So response is referring to how well a patient's disease is responding to treatment. So it means capturing information about whether tumor burden has decreased in size, stayed the same, or gotten bigger. So at the point of care, patients and clinicians really make treatment decisions based on what's happening with the patient's level of response. And also in research, this is critically important. So you'll see commonly response rate being described. So that's a very common outcome used to measure the proportion of patients who are responding to a given therapy. And understanding response in real-world patients like we're able to with Flatiron data really gives us important insights into how disease behaves in routine clinical care and amongst broader populations of patients versus those typically included in clinical trials. So it's able to give us insights into things like identifying patterns of patients who may have a specific clinical or disease characteristic that makes them more or less prone to responding well to treatment. And those types of insights are really helpful in terms of informing treatment decisions and furthering drug development.
Can you explain why machine learning techniques are being used to extract real-world response data from clinical notes and some of the key advantages of using deep learning NLP model over perhaps traditional manual abstraction methods in this context?
Yeah, for sure. So, artificial intelligence has really been transformative the last few years and continues to rapidly evolve. We are able with AI to unlock a scale and level of insight that was previously not possible. So really allowing us to get closer to learning from more patients, really unlocking a scale of insight that we didn't have before. So we've been capturing real-world response data for years using manual abstraction, humans who go into the chart following policies and procedures and capture this information. But that process takes a tremendous amount of time and effort. So you can imagine, in a given patient, response is getting assessed every few months. So going into the chart and curating that data over the course of years of a patient's cancer journey is just a really time-intensive activity. So historically, in order to maintain quality and timeliness in terms of generating that data, we've really had to restrict our ability. We've only been able to generate that type of data on hundreds of patients in terms of cohort size. Now that we've begun to leverage ML and AI, we're talking about hundreds of thousands of patients that we can generate that data on extremely quickly. So it's really changed the game. We're able to leverage this new technology to unlock this valuable data on a much larger scale than we were able to before.
And coming to this particular piece of research, what were the main objectives of the study being presented at ISPOR 2025?
We developed this real-world response approach using ML. We call it scaled real-world response. It's using a natural language deep learning model. And the aim of the study was to describe the reliability, completeness, and internal validity of this approach across 14 of the most common solid tumor cancer types, really in order to establish confidence in this approach and its use for research.
How did you approach the training and validating of the model across such a diverse range of solid tumor cohorts, and what steps did you take to ensure the completeness and internal validity of its outputs?
Yeah, this is such an important question. It's one we grapple with consistently as we are learning and using ML in different ways. This question of how do you know if the performance you see in one cohort with one model can transfer that model's performance to another cohort. So here, we had the foundation of knowledge from our existing work with abstracting real-world response, and I'll say that foundation has been extremely helpful in informing some of our initial design decisions as we approach using ML. So, for example, for our human abstracted approach for real-world response, we've been able to develop an approach that consistently performs well across solid tumors where imaging is the primary modality of assessment. So, we have one set of policies and procedures that we're able to give abstractors to use across those tumor types, and we see consistent performance there. So having that knowledge and having done that with humans, we went into our initial design decisions with the ML approach with a similar assumption. And with my clinical hat on, when I think about the model type we're using here, so a natural language processing model, really what comes to mind for me is how consistent do I expect the clinician's language to be across these different cohorts? So is there reason to believe that a clinician, will document the words they're using to document type of response? So things like complete response, partial response, stable disease. Do we think that type of documentation will be consistent across these cohorts, or could it vary a lot? And really my hypothesis going in was that it would be pretty consistent. The approach to using imaging and describing response is pretty similar across solid tumors. I will note that we do not feel the same about non-Hodgkin's lymphomas. So there are some lymphomas where imaging is used to assess response. But there, because of the dynamics of lymphoma and the types of imaging used, our hypothesis, and we've seen, is that the language used by clinicians is a little different. So we will use a different model there. We won't use the same one that we have been using for solid tumors. So with all that sort of knowledge under our belt, we went into this under those assumptions and attempted to develop a model that would be transferable across those solid tumors. Of course, we then had to test it. And that's where the questions about validity come in. So we were able to look at performance overall and by individual tumor types. We also were able to look at it. There were a few other ways. We were wondering if performance might differ by therapy class being used, targeted therapies versus immunotherapies, or even by line numbers, whether earlier lines versus later lines performance might differ. So those were the different ways we looked at it. One thing I'll add, we were able to train the model using labeled abstraction data from a heterogeneous group of solid tumors. So the model was trained on labeled data from different solid tumor types. And then, into your question of how we looked at completeness and internal validity. So we looked at completeness by looking at the number of assessments overall and the frequency of those assessments. So whether those aligned with clinical expectations, do we see patients getting imaging and response assessments within a cadence that we would expect clinically? or are we seeing, over frequent capture or under capture? And those did align with clinical expectations. In terms of internal validity, this was pretty cool. We used the response data to define patients as responders or non-responders. So patients whose disease was responding versus those who weren't. And as you would expect, we saw patients who were responders had better overall survival across different tumor types and line numbers. So that's how we looked at internal validity and completeness.
What were the key findings of the study?
Yeah, so there are 3 key findings I'd love to highlight. The first is within our initial training and testing, we saw very high correlation, 0.86, between human abstracted real-world response rate and ML-generated real-world response rate. So with response rate, we knew being like the primary way that a lot of researchers would use this data, that was really important to see. Then across these broad disease-based cohorts, which ranged in size from 4,000 upwards of 120,000, we saw rates and timing of assessments aligned with clinical expectations. So about 60 to 80% of patients had at least one assessment within a given line of therapy. And we saw median time to first assessment, second assessments, third assessments was what we would expect. And then, as I mentioned previously, across the most frequent regimens from first line to third line for each disease, we saw a statistically significant difference in overall survival for patients classified as responders versus non-responders. So really overall, we found that the ML approach to generating this response data performed quite well. It was reliable, complete, and then demonstrated internal validity across the tumor types and different line numbers. And it could be efficiently scaled across such large cohorts.
Could you talk a little bit about some of the key lessons learned in scaling this model across the Flatiron electronic health record-derived database and the implications that has for future machine learning applications in real-world evidence?
Yeah, a few things come to mind for me here. I mean, we had so many learnings. One I'd love to highlight is that close collaboration of different functions in a cross-functional team is really crucial, and a willingness to learn from one another. So the technology of machine learning is rapidly evolving. I really rely heavily on my engineering colleagues to translate it into language I can understand. The same way, conversely, they rely on me to translate clinical and oncology lingo into language they can understand. So really coming together in order to make informed decisions to guide model design, anticipate challenges, and also design the meaningful measures of how to assess performance, knowing how the data will be used and how to interpret results has been really crucial. So I'm really, really grateful to work in teams that support that. I'll also mention, the foundational knowledge we have generated in terms of abstracting data with human abstraction really has been valuable for building out our ML approaches. So not only from the practicality of having labeled data that we can train and test the model on, but having the knowledge of what it takes for a human to go in and generate this data, providing policies and procedures for them. The policies and procedures we provide for them are very similar to what we've started using to design prompts for large language models, for example. And the other thing I'll mention, the pace of change here is really something we've had to adjust to. This approach used a deep learning model. We're now on to experimenting with large language models, really learning where the different types of ML tools can best be utilized, what model architecture best aligns with different use cases and tasks we want it to do. For example, some of my colleagues recently published a paper demonstrating how to best fine-tune large language models to accurately extract details about PD-L1 biomarker testing. However, things are moving really quickly. So, that publication came out in January and we're already on to new approaches of how to do that. So I'm interested to see at ISPOR how people are sharing about ML and how people are learning along the way.
How can this validated model be applied by other researchers working with electronic health record oncology data?
Yeah, so I mean, certainly the learnings I've mentioned, I think would be valuable to apply and know in any type of setting using EHR data, or maybe even some other types of real world data. In terms of the model itself, one of the advantages of it is trained on a pretty diverse data set. So the Flatiron Health Network represents, 5 million patients across the US. This was trained on the US data. It spans hundreds of practice sites, includes data generated from both community and oncology centers, and even uses different electronic health records. That being said, it would be important, if transferring this model to another data source, it would be important to validate it in that new setting. But I would think some of the validation exercises that we shared here could be done and generated in that new data source.
Perfect, thank you. So thank you, Kelly. Thank you for speaking to us today. As you say, this is a really hot topic, fast-moving topic, and I think it will be fascinating to see how it develops into the future. So I'd just like to close by thanking our viewers for watching this interview on the evidence space, and thank you again, Kelly, for speaking to us.